So today we're very excited to have Dr. Lowri Allen with us. She's going to be speaking about maternal type 1 diabetes and relative protection against offspring transmission. She's a clinical and academic trainee in diabetes and endocrinology in South Wales, UK. During her medical training at the University of Edinburgh, she completed an intercalated degree in pharmacology, and after graduating with honors in 2013, she completed academic foundation training in Southeast Scotland before moving to South Wales in 2015. As an academic core medical trainee, she was awarded a grant from the Diabetes Research Wellness Foundation, which she used to establish an e-cohort of just under 200,000 pregnancies in Wales to describe pregnancy outcomes in type 1 diabetes as compared with pregnancy outcomes in women without type 1 diabetes. She was subsequently appointed to the WCAT, which is the Wales Clinical Academic Track Program, which allowed her to explore further her interest in pregnancy in type 1 diabetes, and in particular to describe outcomes in teenage pregnancies in type 1 diabetes. Dr. Allen is currently undertaking a PhD as part of the prestigious Wellcome Trust funded GW4CAP PhD program for healthcare professionals. That's amazing. Through her PhD, she hopes to better understand the combination of genetics and intrauterine environmental risk for offspring during those affected by type 1 diabetes developing T1D themselves during childhood. Oh, here's someone joining. Thank you so much for joining us, and we're really excited to hear more about the talk. I'm going to ask that people drop questions during into the chat, and we'll take those questions at the very end. I've also dropped a link to her recent paper for those who have interest.
Great, thank you very much. So thank you for the lovely introduction and for the opportunity to share some of my work today. So as you've heard, my name's Lowri Allen, and my background is as a clinician working in diabetes and endocrinology in the UK. But at the moment, I'm halfway through studying towards a PhD through the Wellcome funded GW4CAP programme for health professionals, which is based in the southwest of England and Wales. So I work at Cardiff University with Colin Diane and Peter Taylor, but I'm also really lucky to be working with colleagues in Bristol and Exeter under the supervision of Kathleen Gillespie and Richard Oram. And as you've heard, the focus really for my PhD is around this concept that you seem to be less likely to get type 1 diabetes if you have an affected mother as compared with an affected father. And so my plan for this session was to talk a little bit about the background to this particular subject matter, highlight kind of what is known, but importantly, what we don't know, share some of the early results from my PhD, and then talk about how this then sets me up to move forward for the remaining time I have left on the PhD and beyond. So by way of background, we know that type 1 diabetes is the end product of a combination of a genetic susceptibility to the disease and then exposure to particular environmental risk factors. And because of this genetic component, we know that your risk of the disease is higher if you have a family history. And so we would often quote that your risk of type 1 diabetes is somewhere between 8 and 15 times higher than the background population if you have an affected first-degree relative, so mother, father, or sibling. But what I'm interested in is the fact that it seems to really matter who that affected relative is. So this study has been published for 40 years now, and it comes from Warren and colleagues from the Joslin Diabetes Center in the United States. And what they did was looked at 419 offspring of men and women with type 1 diabetes to decide to explain what proportion of these went on to develop type 1 diabetes themselves in time. And what they reported was that by the age of 20, 4 times as many of the children of the fathers with type 1 diabetes had developed type 1 diabetes as compared with the mothers. Now, this was quite a small and simple study, but a really important study because it was the first time that anyone had suggested that there was a difference in the risk of type 1 diabetes between those with affected mothers and fathers. And also it's then paved the way for many more research studies in this particular field. So on this figure here, I've tried to summarize the results from all of the key studies that have been published in this field since then. At the bottom, there's a link to, there's a reference to a review article that we published in the Lancet Diabetes and Endocrinology last year, where we look at all of these studies in quite a bit of detail and discuss their strengths and weaknesses. But really, what I wanted to do for now is just to highlight their key results. So you'll see that I've split the studies into 2 different groups. The 4 studies on the top are designed in a way that they take the offspring of men and women with type 1 diabetes and look to see what proportion develop type 1 diabetes over time. So the results you're looking at are odds ratios, where that represents the odds of a child of a man with type 1 diabetes developing the disease as compared to the odds of a child of a woman with type 1 diabetes going on to develop the disease. Then on the bottom, you've got studies where, if you like, they've taken the reverse approach. So here what we have are people who all have type 1 diabetes, and we're comparing the proportion with affected fathers versus affected mothers. And to put the technicalities to one side, the real key message to take away from this slide is that there is consistent evidence that you seem to be twice as likely to get type 1 diabetes if you have an affected father as compared with an affected mother. Let's try and put that in a little bit of context in a way that you might be able to use when talking to patients in clinical practice. So the absolute numbers will depend on where you work and the population you serve, but these are the sorts of numbers that we see locally to where I'm based. So if someone comes to your clinic who's got one affected first-degree relative who is their father, we'd quote them a lifetime risk of type 1 diabetes of about 4 to 6%. For somebody with one affected first-degree relative who is their mother, we would quote a risk of about half of that at 2 to 3%. Studies where they've looked at the risk of type 1 diabetes in people with affected siblings suggest that they fall pretty close to the risk that we see in those with affected fathers. And the reason for highlighting this in the context of this talk is because it means that we believe that it's those with affected mothers that are at a lower than expected risk of the disease, as opposed to it being the case that having an affected father puts you at a particularly elevated risk. Of course, when we talk about a lower risk in people with affected mothers, that's a relatively lower risk, specifically as compared to people with other affected first-degree relatives, because for people who have no affected first-degree relatives, the risk would be much lower. So we'd maybe quote 0.3% lifetime risk locally to us on that basis. But nonetheless, this is a significant difference between the offspring of men and women, and that's both statistically and clinically significant and relevant. You've already seen this figure once, but this time I've annotated it just to give you a sense of the follow-up periods for these studies. And the key thing that I want to highlight is that these studies were predominantly designed to look at the risk of offspring of men and women with type 1 diabetes developing the disease early in life, so during childhood and early adult life. So you can see they're kind of looking at up to age 15, 20, 30. There's just the one study where they've looked at people over the age of 35, but that's really quite a small number of individuals. Why does that matter? Well, it's because up until this point, it's meant that it's been unclear, is this phenomenon of relative maternal protection against type 1 diabetes in offspring as compared to paternal disease quite a short-lived phenomenon, where effectively what you see is that having an affected mother is pushing your diagnosis back later into life than having an affected father. Now, that's still beneficial because we know that developing your type 1 diabetes later in life is associated with improved cardiovascular outcomes, better survival. But clearly for those for those individuals with affected mothers, it would be even better for them if this was a long-term or even a lifelong relative protection that they were getting. Another really important key unknown is, why does this happen? What is the underlying mechanism? So again, this is another figure from that review that I'd mentioned earlier, where we've tried to summarize some of the plausible mechanisms that have been considered to date. Broadly speaking, most of these can be subdivided into those that are predominantly genetic mechanisms and those that are more related to the environment. If we think a little bit about the genetics of type 1 diabetes to start with, we know that most of your inherited risk of the disease is inherited in an autosomal way. So you'd expect the genetic contribution from a mother or a father with type 1 diabetes to their child's susceptibility to be fairly comparable. The caveat to that would be if there was a parent of origin effect at play. So you might find that the risk of inheriting a particular gene is comparable, whether it's your mum or dad who carries it, but the likelihood of expressing its phenotypic effect depends on which parent you inherit it from. Now, in the context of type 1 diabetes, there have been studies that have specifically looked for evidence of a parent of origin effect, and that's included 2 really large studies, one being a large GWAS. And they concluded that it doesn't seem to be a major player in the heritability of type 1 diabetes. And even if there is a small parent of origin effect at play, by itself, it wouldn't be sufficient to account for the magnitude of the difference of risk that we see in terms of type 1 diabetes risk in offspring of affected men and women. So for that reason, there's been more and more focus on the environment and the role it plays in trying to explain this difference in risk. At a really simplistic level, I guess one of the most obvious differences in the environment of a child with an affected mother versus father is that many of those with affected mothers will have been exposed to mum's type 1 diabetes in utero. That means potential exposure to maternal hyperglycemia, insulin therapy, a host of antibodies, both maternal islet autoantibodies, but also other potentially relevant antibodies. So one example that I've noted there would be antibodies against enterovirus, which we know is a potential trigger for type 1 diabetes in susceptible people. And these different environmental exposures could either be having a direct effect on how the pancreas or the immune system is developing and maturing, and that then has a knock-on effect for type 1 susceptibility in that child, or there could be a more indirect mechanism, so for example, an epigenetic mechanism that links these up. Finally, you'll see on this figure that I've also mentioned maternal microchimerism. So this is the idea that some of the maternal cells enter into the fetal circulation during pregnancy. Those cells become recognized by the fetus as being part of its own makeup. And you can imagine that if those cells have come from a mother with type 1 diabetes, that could then have a knock-on effect for that child's type 1 diabetes susceptibility in the longer term. So a range of different plausible mechanisms, and there's lots of work to be done in trying to tease out which of these mechanisms, if any, might explain this phenomenon of relative maternal protection.
Lowri, can I just ask a quick question? Have they examined birth order in this context, in terms of the, those with the maternal type 1 diabetic, influence, or mother with a type 1 diabetes? Is there any, are there any, powered studies to look at that?
So not in this context specifically. They've looked at birth order in general and risk of type 1 diabetes in children, but not specifically, to my knowledge, looking particularly at children who've been exposed to type 1 diabetes. And similarly, there's not a huge amount understood about the relative importance of the duration of the mother's type 1 diabetes, which I guess is partly what birth order would help you to untease.
Mm-hmm. Yeah, it's interesting. Thanks.
No worries. So in terms of moving on to what I've been doing as part of my PhD, the early part of my PhD aimed to address kind of three main areas, really. Firstly, to determine whether this concept of relative maternal protection against the type 1 diabetes in offspring, as compared to paternal disease, is a short-lived effect that is predominantly seen in childhood, or whether it's a longer-term effect that is still evident into adult life. Secondly, to determine whether it's dependent on the mother being diagnosed with type 1 diabetes before the birth of the offspring, because that might then lead weight to any argument that it's in utero exposure to type 1 diabetes that's really critical here. And then finally, we wanted to compare genetic susceptibility to type 1 diabetes in individuals with the disease based on whether they had affected fathers versus mothers. And to do this, we use the GRS2, so a polygenic risk score that's well validated and looks at over 60 different genes known to be highly associated with type 1 diabetes susceptibility, and then uses that information at an individual level to predict how likely someone is to develop the disease based on their genetics alone. So to do this work, we use data from 5 existing type 1 diabetes cohort studies, and I'll just briefly tell you a little about these. So first in the green, we have BOX. So this is the Barts Oxford family study that's conducted in the southwest of England. So it's been ongoing since 1985, and it involves the recruitment of individuals with type 1 diabetes diagnosed before age 21 alongside their family members, and then they're followed up in the long term. BDD stands for the Better Diabetes Diagnosis Cohort. So this is a Swedish national cohort ongoing since 2005 and involves the recruitment of people with type 1 diabetes diagnosed before the age of 18. And since 2011, they believe that they've captured over 95% of their target population at a national level. The T1DGC stands for the Type 1 Diabetes Genetic Consortium. So this is an international research network aimed at recruiting people with type 1 diabetes into research studies, predominantly focused on trying to understand the genetics of the disease. And as part of this network, between 2004 and 2008, a specific families dataset was compiled, which predominantly involved recruitment of affected sibling pairs with type 1 diabetes, but also some affected parent-child trios. So this is a cohort that, if you like, is enriched for people with a positive family history, albeit in most cases a sibling who's affected. TrialNet Pathway to Prevention is another international research network, this time though recruiting not people with type 1 diabetes, but people at high risk of the disease on the basis of their family history and autoantibody profile. So again, enriched for people with a positive family history. And then as part of the research programme, they are then followed up to identify which individuals develop islet cell autoimmunity and who develops clinical type 1 diabetes over time. And then the final cohort is Start Right. So this is a British multi-centre study. And this one is a study that recruits people with adult onset diabetes and is enriched for people diagnosed after the age of 50. So a really valuable source of information on people with adult onset type 1 diabetes. So across these 5 cohorts, people were eligible for inclusion in the analysis for our work if they had confirmed type 1 diabetes and their family history data had been recorded. So across the 5 cohorts, we ended up with 11,475 participants that we could include in the study, making it the largest study in this field to date. But most importantly, these were people diagnosed with type 1 diabetes anywhere between the age of 0 and 88 years of age. So it's the study with the broadest age range of participants at the time of type 1 diabetes diagnosis, meaning they were fairly uniquely placed to comment on how this phenomenon of relative maternal protection might apply to people in adult life.
Can I just interrupt one more time? Can you tell me how challenging was it to assemble, to access first, and then to assemble all these cohorts?
Yeah, so we're lucky in the type 1 diabetes world that I think we've got very good collaborative networks that mean that identifying the cohorts wasn't too challenging. But as always, there's lots of kind of administrative processes in terms of trying to access the data. And then also they're all a little bit different. So one of the key things in the analysis was, because they're all different, rather than lump them all into one analysis, what we've done, and you'll see this as I move through the slides, is that we've analysed within each cohort separately to start with, and then we've used random effects meta-analyses to derive overall estimates of effects across the different cohorts. So I think there are definitely challenges to working across cohorts, but I think the real strength of being able to do that is that you do cover so many different populations across different parts of the world, but also it's meant that we've got children, adults diagnosed with type 1 diabetes, and I think that comparison was really important to move forward what we understand in this field. And I don't think there would have been one study that would have given us enough people across that broad age spectrum.
Yeah, no, this is a big lift. I mean, this is really, really amazing what you've put together here. So I just wanted to acknowledge that.
Thank you. So moving on to what we found then, so as I mentioned, as we go through the results, what you'll see is results for individual cohorts and then an overall estimate of effect at the bottom from a meta-analysis model. So the first thing we did really simply was looked at across these cohorts and said, of these people with type 1 diabetes, what proportion have an affected father versus mother? And as you'd expect, based on the background literature that I've presented so far, what we found is that you were about twice as likely to have an affected father versus mother. But just to focus in on 2 of these cohorts for a moment, so you'll recall that I said that BDD was people who were diagnosed before the age of 18, whereas Start Right is our exclusively adult onset cohort. And it was really striking to us that the point estimate was really quite similar for these 2 different cohorts. So what we then did was we subdivided all of the participants across all 5 cohorts according to whether they were diagnosed with type 1 diabetes as adults or children, using age 18 as the cutoff. And what we found is that in both of those subgroups, you were significantly more likely to have an affected father as compared with an affected mother. So in this figure, I've shown the detailed results for those who were diagnosed as adults at over age 18, and you can see you're significantly more likely to have a father with the disease than a mother with the disease, even if you're not diagnosed until your adult life. Another way of kind of looking at this issue around the longevity of any relative maternal protection was to compare age at onset of type 1 diabetes for those with affected mothers and fathers. So the rationale here is if having an affected mum is just giving you short-term protection, then your age at onset might be older compared to those with affected fathers. But actually, we found there to be no significant difference in age at onset in those with affected mothers and fathers across any of the cohorts. And to look at that just in a little bit more granularity, we took the individual patient level data from all 5 cohorts and generated these cohort stratified Kaplan-Meier curves that are indicating type 1 diabetes-free survival time. And the key thing to take away from this figure is that the curves for those with affected mothers and fathers are almost identical, so further refuting the idea that we're seeing delayed disease development in those with affected mothers. So I hope that those kind of slides that I've shown so far will have convinced you that not only do we see this relative protection against type 1 diabetes in those with affected mothers versus fathers, but that this is a long-term effect that we still see into adult life. Did we get any further insight into what the mechanism might be? Well, for 2 of the 5 cohorts, we knew exactly when the parents had been diagnosed with type 1 diabetes. So that meant for children with affected parents, we could subdivide them based on whether their parents were diagnosed before or after they were born. So if we focus in first on people with parents diagnosed before they're born, we see that there's twice as many affected with affected fathers as compared with affected mothers. But if we look at parents diagnosed after the birth of their offspring, we find that there's no longer any significant difference in the proportion of affected fathers and mothers. So what we're saying is that in our cohort, we're only seeing evidence of this relative maternal protection when parental disease is diagnosed before the birth of the child in question. And that would strengthen the argument that in utero exposure to type 1 diabetes might be a really important driver of this mechanism. The final thing we wanted to look at was to compare the GRS2 as this marker of inherited genetic susceptibility in participants with affected mothers and fathers, and we found that there was no significant difference in these 2 subgroups at all. What that implies to us is that the genes that you inherit don't seem to be significantly different in terms of their quantity between those with affected mothers and fathers. It doesn't exclude the possibility that the expression of the genes that you inherit might be different, as that's not measurable by the GRS2. But as we've already alluded to, there's literature out there that suggests that a parent of origin effect is not a main driver of this mechanism. This also doesn't exclude the possibility that any environmental exposure or any in utero exposure specifically could be exerting its effect through an epigenetic mechanism. But what can we say? Well, I hope you will be convinced that there is consistent evidence of a long-term relative maternal protection against type 1 diabetes in offspring as compared with paternal disease. In our cohort, this was dependent on parental disease being diagnosed before the birth of the child in question, and that coupled with the findings from the GRS2 analysis lead us to believe that in utero exposure to maternal type 1 diabetes is likely to be really important here. Regardless of what that exposure might be, given that it results in such a long-term relative protection, it must be resulting in a permanent modification of some aspect of the child's susceptibility to type 1 diabetes. So where do we go now? Well, what we really would like to do is really unpick what exactly is going on, what is the mechanism, and in particular, what happens in utero to a child whose mother has type 1 diabetes that might influence their long-term susceptibility to the disease. And we're particularly focused on understanding the relative importance of exposure to hyperglycemia in this context. So what I want to explore is, does the severity of the maternal hyperglycemia during pregnancy have implications for the long-term risk of type 1 diabetes in the child? But also, can we learn anything from exposure to hyperglycemia in other contexts? So what if you're exposed to hyperglycemia because your mother has type 2 diabetes or gestational diabetes? Do we still see some element of protection, or is this something that's really specific to the autoimmune-driven hyperglycemia of type 1 diabetes? The ultimate ambition here is that if we can understand exactly what the mechanism is, that might then open up new research avenues where we try and develop therapeutic measures that replicate the protective elements of maternal type 1 diabetes in a way that could reduce or prevent type 1 diabetes in people who are particularly vulnerable to the disease. So I'm going to stop there. There are many, many people who've inputted into this work who is appropriate for me to acknowledge and thank, but I welcome any questions.
Well, I would just say that, yeah, again, this is just a huge body of work that you've, been able to unearth here and really subject to analysis. And you have such a great team there, at Cardiff. It's really amazing how much is being done at that institution. So kudos on that front. I wanted to ask you a couple of things regarding the hyperglycemia and its fueling of the risk of T1D. So, some literature also suggests that during pregnancy is when women have, very good control.
Yeah.
Because the fetus basically, or the developing fetus is sort of siphoning off extra, glucose or higher glucose. And, then if there is poor control, the developing infant, becomes larger, and then you can have a large birth. So what about this whole idea of the fact that maybe they're really in great control during the pregnancy, and that has some kind of influence. Is there any thoughts around that?
Yeah, so absolutely. So this is really interesting and a fairly challenging area to address, because we spend all of our time in the antenatal clinic telling women it's really important to have good control in pregnancy because it improves outcomes. But in this context, what we're actually wondering is whether, in order to get this relative maternal protection, you actually need to be exposed to a higher level
Yeah.
of hypoglycemia, because
Yeah.
that's what then drives the pancreatic development in the child. And then that makes their pancreas more robust to withstand the kind of tests, if you like, that someone who's susceptible to type 1 diabetes might go through. So that's what we really want to understand is it actually those children who are exposed to the very highest levels of hypoglycemia in pregnancy that are actually being protected? And if that is the case, what does that mean in a clinical setting? Because clearly we're not going to go around telling pregnant women, let your blood sugars run high, because there's lots of other downsides to that. But the question might be, could there be something that we could do therapeutically that might have a similar effect, but without driving high blood glucose? So is there something that specifically is involved in modulating kind of islet cell function that we can target that has the same end result, but without kind of the complications associated with hypoglycemia in pregnancy?
Yeah, I mean, I guess you could get at, like, who is really experiencing hyperglycemia with CGM data,
Yeah.
but maybe you could even go after sort of a retrospective analysis using who ended up with high birth weight. Um,
So that's kind of the next chapter of the PhD. Is at the moment we're using Welsh data, but also Swedish data to kind of take that epidemiological approach, which has its own challenges, because although CGM is something we use a lot now, kind of looking back at cohorts over time, we don't have that granularity of data on glycemic control in pregnancy. And so that's hopefully something we'll get more of as time goes forward. But there are also other kind of markers that we're looking at that you might be able to use as surrogates of glycemic control in pregnancy that we hope will kind of help boost that approach. But that's kind of what I hope. I might be able to come back in a year or so with more information on that. But I think that's certainly where we're going next with this work.
Yeah, no, you've achieved so much in such a short time. So yeah, that would be fantastic. I wondered, what about the Australian data sets? Have you thought about, sort of wading into those or incorporating them? I know it's already, pretty tough to have the standardized, harmonized set you've got, but they do have quite a bit of data.
So I'm actually really lucky that one of my collaborators for my PhD is Claire Meek, who's working on India data. And we've been setting up opportunities to collaborate using some of the India data alongside concept data from the UK, which will be a really exciting opportunity because
Yes.
they've got that level of data on glycemic control in pregnancy and lots of different measures around that a lot of other cohorts don't have. So that's the challenge, really, is looking at what each cohort brings and how can we look at this question from as many different angles as possible to really try and hone in on what is the exact mechanism that underpins this.
Yeah, no, that will be, oh, that's really exciting. I'm so excited. I'm so glad to hear that. Yeah, India has a wealth of information. Would anyone like to unmute or raise their hand and ask a question? See, Stephanie's here. Okay, I'll ask a few more then. I also wondered, what are your thoughts around this sort of, can we unpack a little bit more the sibling data? If you're, a sibling of a type 1 and that type 1 is either male or female, is that sibling's risk for a child impacted?
So
And you
yeah,
kind of touched on it.
So I think some of the Swedish data that we're going to look at will hopefully allow us to do a lot of sibling comparator work, because they've got a huge amount of kind of that family structure work that we're hoping will allow us to look at this. But I think alongside kind of looking directly at sibling comparisons, I think what we also really need to do is understand this relationship between the timing of when mum's diagnosed relative to the child's birth, her age at the time that she's diagnosed with type 1 diabetes, and the duration of her diabetes at the time the child is born. Because it could be that actually what we're seeing is that the timing of diagnosis is actually acting as a surrogate marker for age of diagnosis or duration of diagnosis or something in the mother. And in this analysis, we didn't have enough granularity of all those different bits to look at the interaction between the three. But I'd really like to try and unpick how they all influence each other and what is the most important component of that. And as you say, I think adding the sibling comparator in might also help to untease that, because you'll see, the duration will get longer with the birth order, etc.
Yeah, no, you've got so many interesting avenues going here. It really is exciting. We do have a question from Steph Hannah. I wondered what your thoughts were on Randy Johnson's work on DNA methylation differences between people with mothers or fathers with T1D. That's really a great question.
Thanks, Steph. So I think I'm probably out of my comfort zone a little bit talking about kind of epigenetics, which is outside of my expertise. But I think it's a really important area that we need to be looking at in this context, because I think whatever's happening in utero, there's a high chance that that could be leading to kind of epigenetic changes, and that could be the kind of missing link here. So I think the key thing is to work with colleagues that are taking these different approaches so that ultimately we can try and unpick the mechanism, because I think one study is never going to find the answer here. And actually combining this more epidemiological approach with looking at the epigenetics, I think will make a big difference to our understanding of this field.
Yeah, that's a very new sort of frontier. And I think it will really merge nicely with these data once they're out and once they're fully and analyzed. I wondered also, in sort of that same vein, have or is there a data set that looks at sort of viral insult in terms of COVID, SARS-CoV-2, for mothers who went through that pandemic, and whether or not that impacted that protection that they are showing?
That's a really interesting question, and I don't know the answer to that. Actually, I'm not aware of a kind of study that's ongoing in that particular field, but certainly there's been, various groups are interested in viruses in more broadly, and particularly one of the kind of questions has been, is some of this to do with actually enterovirus? And actually it might be that a key thing when we're trying to unpick the kind of idea of duration of type 1 diabetes, could it be that actually one of the things here is that the longer your mum's had diabetes, the longer it's been since her exposure to potential triggers, including enterovirus, and that her antibodies to enterovirus might wane, and that could then affect what she passes on to her child. And then the child's susceptibility to enterovirus as a trigger could be partly explaining this. So I think there's certainly lots of work to be done looking at the kind of virology and the viral aspects of this, but I'm not aware of any work ongoing currently in the field of COVID specifically, but that's not to say there isn't.
It seems like it could be a low-hanging fruit.
Mm.
And then I guess my last question, in the interest of time, because I've been asking a lot, is what are there any data around microbiome sampling in these cohorts?
So I think there's lots of work ongoing aimed at looking at the microbiome. I don't think there's a huge amount of literature that's published yet, but I think there's a lot of work being done that means that that literature will become very enriched in the next few years, because certainly that's something that there's been a lot of interest in the UK and around the world.
Yeah, just wondering if those studies themselves have these data sort of like embedded in them, and it just needs to be, again, teased out and reanalyzed.
Yeah, I think the challenge is that having cohorts where we've got really accurate family history data, and that data of what happened during the pregnancy, but also what's happened to the offspring during follow-up. And that's what's we found most challenging in putting this work together, which is why sometimes I think what we need is lots of people to come at this from different angles, because different studies will cover different aspects of it to a greater or lesser degree.
Yeah, absolutely agree. The complexity of the disease really calls for an interdisciplinary lenses to examine it, and bring their expertise to bear in interpretation. So thank you again, Lowri. This was really, really interesting, and we can't wait to see what comes next from your next stage of analyses. It's really, really an exciting time in terms of these data. And we'll definitely be watching for your next papers that are coming out, and or next presentations. Thanks again for joining us.
Thank you very much for the opportunity.
Have a great rest of your day. Bye-bye now.
And you, bye.